Hello to everyone, I'm Ileana Tossolini and I'm going to talk about our findings on co-transcriptional microRNA biogenesis in plants and the essential role of R-loop hybrids between the nascent pri-miRNAs and the encoding loci. In plants, microRNAs are a class of 20 to 24 nucleotides small RNAs that repress gene expression at post-transcriptional levels. Plant microRNA genes are transcribed by RNA polymerase II as capped and polyadenylated primary transcripts known as pri-miRNAs, which are highly variable in length and secondary structure. The biogenesis of microRNAs in plants is a unique and evolutionary divergent pathway, and unlike the animal pathway where Drosha and Dicer concatenate to produce the mature microRNAs, in plants a single processing complex formed by Dicer-like 1 and accessory proteins leads the entire process inside the nucleus. And the characteristics of each prime microRNA define the biogenesis way. So alternative processing directions can go either from the hairpin base to the loop or from the loop to the base. And also Dicer can produce sequential cuts on prime microRNAs every 21 nucleotides to release the mature microRNA. Recent studies have shown that some processing complex proteins, such as DCL1 and HIL1, associate with microRNA loci, besides their canonical locations in the bodies. And it opens the question of whether such a recruitment induces co-transcriptional processing or only allows an earlier complex assembly. So, answering whether plants' prime microRNAs can be co-transcriptional processed is one of the most outstanding open questions in the pathway. Here, we used plant-native elongating transcript sequencing, known as PlanetSeq, to profile genome-wide processing intermediates of nascent prime microRNAs that are associated with the RNA pod 2. So, what do we expect to see when plotting PlanetSeq data? Basically a peak where Dicer produced a cut on the prime microRNA. And in order to visualize the general processing profile, prime microRNAs loci were scaled from microRNA 5' to the microRNA 3' to a fixed length. Also, prime microRNAs were sorted depending on their processing direction, base to the loop, loop to the base, either simple or sequential. So, in the case of the prime microRNAs processed from the base to the loop, we can observe here a clear and narrow peak corresponding to the nucleotide right before the Dicer cut site. Interestingly, we also see a peak matching the last nucleotide of the microRNA 3' sequence. And this could indicate that the processed hairpin, the so-called pre-microRNA, is retained temporarily in the co-transcriptional processing complex. When we analyzed loop-to-base prime microRNAs, we detected two clear peaks corresponding to the first and the second Dicer cuts. And this indicates that the entire biogenesis progression occurs co-transcriptionally when the prime microRNA is still attached to the locus. And I will talk later about the analysis of this peculiar retention of the 5' end processed fragment. Here is the sequential base-to-loop prime microRNAs global profile, and alike base-to-loop profile, we can see a peak matching the nucleotide right before the first Dicer cleavage site, but in this case, the subsequent sites are not detected. So these profiles support the scenario where only the first processing step of base-to-loop and sequential base-to-loop prime microRNAs takes place co-transcriptionally. If we observe the global profile of sequential loop-to-base prime microRNAs, we can see this beautiful pattern of peaks matching each Dicer processing site. So these results indicate that all processing steps in loop-to-base and sequential loop-to-base prime microRNAs occur co-transcriptionally. We also found a curious result in the global profile of both loop-to-base and sequential loop-to-base prime microRNAs. This additional peak matching the 3' last nucleotide of the microRNA 3'. We found that this peak was only contributed by some prime microRNAs, and when we mapped the entire PlanetSeq reads over their loci, we saw that all reads ending in this precise position correspond to the mature microRNA 3', which are highlighted here in yellow for these particular examples. So this also indicates that some mature microRNAs are temporarily retained at their encoding loci, probably bound to a biogenesis protein such as HIL1 that is known to associate with the mature microRNA after processing. To validate these findings, we first performed an RNA immunoprecipitation using an H3 antibody on cross-linked samples to evaluate processing fragments of pri-microRNAs that are associated with the chromatin. We confirmed what I showed you before. We found both the 3' and 5' ends processing fragments of pri-microRNAs associated with the chromatin, but we observed a relative depletion of the stem loop. Such lack of the hairpin region, which is not evident in the processing defective mutants, is indicating co-transcriptional processing of the pri-microRNAs. We also purified nucleoplasm and chromatin and quantified the abundance of the stem loop relative to the unprocessed pri-microRNAs. And validating our previous analysis, we found that the hairpin region of base-to-loop but not loop-to-base pri-microRNAs was enriched in the nucleoplasm compared to the chromatin. Here I show you our proposed schema for co-transcriptional processing of plant pri-microRNAs. Biogenesis of base-to-loop and sequential base-to-loop pri-microRNAs initiates co-transcriptionally but ends in the nucleoplasm, and all processing steps in loop-to-base and sequential loop-to-base happen co-transcriptionally when pri-microRNAs are still attached to their loci. The detection of co-transcriptional processing does not necessarily imply that all microRNAs are processed exclusively in a co-transcriptional way. So we reanalyzed planet-seq data, taking into account the first dicer cleavage site, indicated in this figure by the letter A. And we calculated the ratio between the reads ending in these sites, that's it, the co-transcriptionally processed reads, and those reads expanding the site corresponding to the unprocessed pri-microRNAs represented here by blue and cyan lines. We used two independent experiments and plotted the ratio for each individual pri-microRNA. Ratios above 1 indicate more co-transcriptional processing. We found that about the 25% of the pri-microRNAs are co-transcriptionally processed, and the 8% showed an equal preference for co- or post-transcriptional processing. And about the 67% of pri-microRNAs seems to be processed more post-transcriptionally. So these results suggest that the co-transcriptional processing of pri-microRNAs is like a dynamic and potentially regulated process, which is not identical for each locus. We next studied if different conditions could affect such balance. So we recalculated the ratio of co-transcriptional to post-transcriptional processing using planet-seq data obtained from plants incubated for 12 hours at 4 degrees and for plants carrying a mutated version of this RNA pol II subunit that accelerates the transcription rate. As we can see here, a reduction of the plant growing temperature impacts the ratio, especially for some individual microRNAs. And with a few exceptions, the increment in RNA pol II speed decreases co-transcriptional processing. Considering that at least the entire stem loop region of a pri-microRNA needs to be transcribed before processing starts, our result may imply that the quick transcription ends up releasing the mature pri-microRNA transcript before it could be co-transcriptionally processed. So, whether a pri-microRNA is processed co- or post-transcriptionally is not a fixed parameter, and this balance can be altered in given conditions. This opens the question of whether a microRNA may have a different function depending on when or where it's processed. One of the most curious results so far was the systematic detection of the processed 5' region of the pri-microRNAs. Similar profiles have been reported, for example, during splicing, where the spliceosome complex stabilizes the messenger RNA fragments. But different from this situation, where each intron can be removed while transcription proceeds, co-transcriptional pri-microRNA biogenesis requires the transcription of at least the entire stem loop region before the processing can start. So it's possible that the slow transcription may assure that the hairpin structure is transcribed and folded, and also may cause the formation of an R loop between the 5' end of a pri-microRNA and its encoding locus. This R loop could stabilize and anchor the pri-miRNA to allow co-transcriptional processing. So we analyzed drip sequencing data searching for the presence and pattern of R loop formation over microRNA loci. We first scaled all pri-miRNAs using either a fixed window from the transcription start site to the most upstream dicer cleavage site, or from this site to the end of the microRNA 3'P. We found frequent R loop formation over these loci, which we also confirmed by drip qPCR on some specific pri-miRNAs. Strikingly, when we ask whether the presence of an R loop impacts co-transcriptional processing, considering the ratio we calculated before, we found that microRNAs with R loops in the initial region of the transcripts are preferentially processed in a co-transcriptional way. On the other hand, pri-miRNAs without R loops over their loci or without the characteristic R loop near the transcription start site showed poor signs of transcriptional processing. Then we analyzed polycistronic microRNAs encoded by a single transcript, and we observed that the microRNA hairpin located adjacent to an R loop was either more efficiently co-transcriptionally processed or even the only one showing signs of co-transcriptional biogenesis. This reinforces our hypothesis that R loop formation between a nascent pri-miRNA and its encoding locus anchors and stabilizes the molecule, allowing the co-transcriptional processing. In conclusion, we confirm that pri-miRNAs are processed co-transcriptionally in Arabidopsis by planet-seq profiling. We also found that once initiated, co-transcriptional processing can occur entirely associated with the transcriptional complex in the case of loop-to-base microRNAs, or in a two-stages fashion for base-to-loop, resembling animals pre-to-pre microRNA processing. We also discovered that co-transcriptional and post-transcriptional processing coexist and fluctuate between growth conditions. Surprisingly, we found that co-transcriptional processing largely relies on R loops hybrids between the nascent transcripts and the microRNA encoding loci to stabilize the pri-miRNAs and allows DCL1 processing. Overall, our study identified an alternative microRNA biogenesis pathway and discovered an unexpected function for R loops promoting RNA processing. This opens the door to new functionalization of co-transcriptionally processed microRNAs that boosts countless regulatory scenarios. Well, I would like to thank you organizers for this opportunity to present our work, and thank you for your attention.